Hello students, this is Inheritance and Variation Part 4. So you can get a question on differentiate between homozygous and heterozygous. Okay, so I have collected some data on this. These are the tall two plants tall. This is tall plant which is pure plant. This is having a capital T, that means it's a pure tall plant. Okay, it's a homozygous plant, and this is a dwarf plant. It carries small t gene which is homozygous. So what is homozygous plant? Homozygous is the presence of two copies of the same allele which codes for a particular trait. So they are having same allele, capital T, small t. So these are the two copies of the same allele they carry. They have either dominant or recessive allele pairs. Example, they will have either capital T, which is supposed to be dominant, small t, it is recessive. You will come to know in monohybrid ratio that why capital T, or that genotype is dominant over small t, that is recessive. So they have either dominant or recessive pairs. Example, capital T or small t. They produce a single type of gamete. It will form or contribute capital T gamete. It will contribute small t gamete, a single type of gamete. Self-breeding produces the same trait over generations. If we self-breed, that is self-pollination. If we take a tall plant which is having the genotype capital T, if we self-breed or self-pollinate the flower or self-pollinate the plant, we'll get the same trait. A pure line will get. In the same case with this pure dwarf plant, we'll get the, after self-breeding, we will get the same trait over generations. Homozygous dominant and homozygous recessive, these are the two types of this homozygous in the homozygous plant. Now, when we cross these two plants which are homozygous, when we cross this capital T tall plant with the small t dwarf plant, when these two pure plants, pure lines, tall plant is crossed with dwarf plant, then we will get this type of heterozygous plant which is having, this is the F1 generation, which is carrying the genes, one from the female plant and one from the male plant, capital T and small t. Okay, so this condition is known as heterozygous condition, where the gametes, they are not similar, they are different. So what is heterozygous condition now, and what are the points? In heterozygous is the presence of two different copies of alleles of a particular gene. So capital T and small t, these are the two different copies of the alleles which are present in that plant, have both dominant and recessive alleles. So capital T represents the dominant character and small t represents the recessive character. So it carries both the genes, okay, or both the alleles. Produce both types of gametes with dominant and recessive alleles. So it produces capital T gamete and small t gamete. So produce two different types of gametes. Self-breeding produces a combination of differences. So self-breeding, it gives the same trait here in homozygous, while in case of the heterozygous condition, different traits are formed. So complete dominance, incomplete dominance and codominance, these are the three types of heterozygous condition. So what is monohybrid ratio? Monohybrid ratio, it is heterozygous for one trait and produced in a cross between two pure parents. If you remember the previous slide where two pure plants were crossed and we got a heterozygous plant, okay, which was carrying capital T and small t. So between two pure parents differing in a single pair of contrasting character, we got hybrid tall in the previous slide, if you remember. So this is the diagram where you can see P stands for parents, parental generation, okay. So female plant and male plant. In these two plants, when the female tall plant was crossed with male dwarf plant, Now capital T, small t, these are the two pure lines, pure traits. Phenotype, genotype, okay? And the gametes which are formed. The tall, pure trait form one type of gamete, that is capital T. Dwarf, pure trait contributes small t type of one type of gamete. In the F1 generation, now what is F1 generation? F1 generation is known as first filial generation. First filial generation, it refers to the, it consists of all the offsprings produced from a parental cross. Alternatively, it is the first generation from a given mating between the pure parents having contrasting characters. Okay, so when we cross the two pure plants, we get the first filial generation. And in this first filial generation, in the monohybrid, all tall plants were observed, recorded. Okay, so when the tall plant is crossed with dwarf plant, no dwarf character was found to be present in the F1 generation, but Mendel got all tall plants. Okay, and the gametes, but that this tall plant is different than this tall plant. Why? Because this tall plant is a pure tall, okay, pure trait, a pure line. It carries both the gametes. It carries both gametes tall, okay, capital T and capital T. There is a difference in the genotypes of these two tall plants. And this F1 generation, the tall plant, it carries capital T and small t. The capital T is, it is replaced by small t, the another gamete of another plant. Okay, but this t is suppressed, it is hidden. Okay, it is not in the observable format. We are not getting a dwarf plant here. Okay, so this is tall plant, but the genotype is different. Even we are getting the same phenotype. So what are the gametes which are formed now from this tall plant? We are getting two types of gametes. This is a capital T and small t. So there is segregation or separation of the gametes. The capital T and small t, even if we are crossing the capital T and small t plants, the gametes which the heterozygous plant is carrying from the parents, the gametes are not getting mixed. Capital T is not getting mixed, but they are in the separated forms. Okay, so we are getting two separate types of gametes, capital T and small t. So there is segregation. Segregation means separation of the gametes. Now what Mendel did, he self crossed the F1 monohybrids. So he F1 generation tall plant was self crossed. Okay, he crossed F1 into F1 and he got F2 generation. Self crossing was done. Okay, self pollination was done, and in this he got this type of result. What is the result? See the gametes, male and female, both the plant are having the same genotype, capital T, small t, capital T and small t. Okay, so when he's after selfing of F1 generation, he got F2 generation, where the result was different. What was the result he got in the F2 generation? That tall plant, there were three tall plant. Okay, now this tall plant is carrying capital T, capital T. These are the two tall plant which are carrying the capital T, small t type of genotype. Okay, this genotypically they are different, but phenotypically three are same. Three are tall, one is dwarf. Okay, so phenotypic ratio is 3 tall plant, 1 dwarf plant. Genotypic ratio is 1 capital T and 2 capital T small t and 1 capital T. So this is phenotypic ratio. Phenotypic ratio is 3:1, 3 tall plant, 1 dwarf plant. Genotypically they were different. Okay, 1 capital T, 2 capital T small t and 1 small t. So he collected the data in a mathematical form by using a checkerboard or Punnett square method. Okay, and he got the result. So if the question is asked in the on monohybrid ratio, it is not compulsory for you to draw the plant, the diagram of the plant. Okay, so the material is important, what you are writing. So you can observe in the next chart. Phenotype, you have to write in this way if the question is asked on the monohybrid ratio. There is no need to draw the tall, but you have to write, we have to specify that which is the tall plant, which is the dwarf plant, and the genotypes. Okay, phenotype is tall when it's crossed with dwarf plant. See, genotype is capital T, small t, which is a pure trait. Gametes, capital T and small t, as they are homozygous. And in the first filial generation or in the F1 generation, the gametes are getting fused from both the pure plants, and we are getting a heterozygous plant which is having phenotypic characteristics same with the tall plant with the parent, but genotypically it is different as capital T and it is having capital T, small t. So of selfing of F1 generation to get the F2 generation, tall is crossed with tall, it is self-crossed, self-pollinated, and he got capital T, small t, capital T, small t. These are the gametes. Okay, so this is the F2 generation where female gametes and male gametes are shown. See the result: capital T, one genotype. Okay, two capital T, small t, and dwarf small t. Okay, that you have to write, and you have to draw this checkerboard. This is very important if you are writing the answer on monohybrid ratio. So let's move to the next slide. So what are the points that Mendel observed in the phenotypic in the monohybrid cross? He did monohybrid crosses with all the 7 pairs of the contrasting characters separately and obtained similar results. In this example, we have concentrated only one character, monohybrid. We considered a single character, that is height of the plant or stem height of the plant. Okay, so Mendel, he crossed all the 7 pairs. We have studied the 7 traits. So he separately studied all the 7 characters and he repeated the experiment ample of times when he studied the monohybrid ratio. Okay, only one of the 2 characters was expressed in F1 generation, and he observed that even if he crossed the 2 pure lines, only one character was expressed in the F1 generation. That even if he crossed tall plant with the dwarf plant, he got only tall plants. Okay, such type of only one character out of 2 characters was expressed in F1 generation. He observed that in F2 generation, the character which was expressed in F1 was in large number. So 3:1 if you observe the ratio. So he got in F1 generation which type of plants he got? He got tall plants. And when he crossed the F1 generations, selfing when he carried out, and he got F2 generation, in F2 generation he got the same character in large number, that is he got 3 tall plants out of 4 and 1 dwarf, that is large number of tall plants. Whatever character was expressed in F1 generation, that character was large in F2 generation, and the other in small number, and the ratio was 3:1, and this ratio is called as monohybrid ratio. Now this point is very important if you are attempting the question on monohybrid ratio. What is monohybrid ratio? That you have to write, and the number, that is 3:1, that you have to write, because it is the characteristic feature of the monohybrid ratio. Moving to the next slide, see how he crossed the different 7 traits. Okay, and he got the same result, that is monohybrid ratio. Tall and dwarf, as we have explained, as I have already explained the tall and dwarf character. F1 tall plant, F2 he got tall in many number, and the ratio is 3:1. Near about ratio is shown here. He crossed again the yellow and green seeds. He got yellow seeds in F1 generation, yellow seeds more in number in F2 generation, green are less. That is 3:1 ratio. If you take another example, violet and white flowers. Violet and white flowers, when they crossed, violet flowers were many in F1 generation, they were expressed, and in F2 generation, violet flowers were many, comparatively white flowers. So all the 7 characteristics, all the 7 traits were crossed, and he experimented with all the 7 traits, and he got the same ratio, that is monohybrid ratio, that is 3:1. So on the basis of that, he put forth, explained the 2 laws. He stated the 2 laws, that law of dominance. Now try to understand this law of dominance. What is it? First law of inheritance, which is law of dominance, is known as first law of inheritance. Okay, so what the law of dominance or first law of inheritance, it expressed in a cross between 2 organisms pure for any pair of contrasting characters. The character that appears in F1 generation is called dominant. So the character which he found or which he observed in the F1 generation, that character, he stated that character as dominant. Okay, and the other which is suppressed is called as recessive. In this law of dominance, he stated that the character which get expressed in F1 generation is called as a dominant character, while the character which gets suppressed in the F1 generation, that character is known as recessive. Thus there is always uniform expression in the F1 generation, that he got in with all the 7 traits in 3:1 ratio. Each character is controlled by a pair of factors, and when it's a pair of dissimilar factors, only one factor get expressed. Thus one is dominant and the other is recessive. So that is a law of dominance. Only one character get expressed in the F1 generation, while the other character remains hidden or it get suppressed. So that is known as law of dominance. While talking about law of segregation now, in the law of segregation, this is the second law which he again stated on the basis of the monohybrid ratio. So law of segregation, law of separation, or second law of inheritance, this is known as first law, law of dominance, that you have to remember. Okay, and law of segregation is known as second law of inheritance. So in this, members of a allelic pair in a hybrid remain together without mixing with each other and separate or segregate during gamete formation. Thus, gametes receive only one of the 2 factors and are pure for a given trait. Therefore, this is also known as law of purity of gametes. So, if you remember the previous, when we have discussed the pure lines, means crossing of the pure lines, if you observe now this, when the pure tall plant was crossed with pure dwarf plant, in this pure tall homozygous is carrying the 2 gametes, similar gametes, capital D, it was crossed with small t, a pure dwarf. Okay, so when these 2 pure plants were crossed, he in F1 generation he got heterozygous plant, 1 gamete from a tall plant, female plant, 1 gamete from a dwarf male plant. Okay, so what he got, that even if the allelic pair is there in a hybrid, they remain together without mixing. So this capital T and small t, they are in the form of separate gametes. Okay, thus gametes receive only one of the 2 factors and are pure for a given trait. Therefore this is also known as law of purity of gametes or law of segregation of gametes. Okay, so that's all about law of dominance and law of segregation. So what are the Mendel's conclusion? By these 2 laws, all the characters inherited separately as discrete particles or units, he termed them as factor. As genes, alleles were not, uh, these terms were not invented at that time. So he inherited characters, whatever they were inherited separately as or by units, he termed those units as factors. Okay, each factor exists in 2 contrasting or alternative forms. For example, for height of plant, there are 2 forms, one for tallness and other for dwarfness. We call them as alleles or allelomorphs. One of the 2 forms is dominant and the other is recessive. Only dominant expresses in F1 generation, and the recessive character it gets suppressed, okay, in the F1 generation. In an organism, inheritance of each character is controlled by a pair of factors. One of the factor is contributed by male, other from a female. So sexually reproducing organisms, the cell is diploid in number, but when we talk about the gamete formation, from the appearance of recessive trait in F1 generation, he concluded that the gametes they don't get mixed, but they just remain together, okay. During gamete formation, these factors they separate or segregate, and each gamete receives only one factor from each pair of the factors. That is, gametes they are haploid, okay. So the gametes they are haploid in case of gamete formation. From a diploid cell we get haploid gametes. So these are the Mendel's conclusion. So hope you have understood the monohybrid ratio. Again, the terms phenotypic ratio, genotypic ratio, what is phenotype, what is genotype, and the monohybrid ratio, that is 3:1, okay, and law of dominance, that is first the characters which are appearing in F1 generation, those characters, these are dominant characters. The characters which get suppressed by the dominant characters in the F1 generation which remain hidden, these characters, these are known as recessive characters, and these recessive characters they remain hidden in the F1 generation, but when we self cross or when we self pollinate the F1 generation, then we there is reappearance of the recessive character in the F2 generation, okay. So the Punnett square, that checkerboard method was introduced by Mendel, and because of the mathematical data, because of the statistical collection of data, whatever he did, so because of that, the law of uh, inheritance, it is the basic fundamental law. These laws they are considered to be basic fundamental laws in the biology. So we have studied the two laws, law of dominance and law of segregation, that how the gametes they don't get mixed, they remain separated, segregated, and they express themselves separated, okay. So hope you understood today's lecture. Do read the matter which is present in your PDF format. It is very important. Exam point of view, this part is very important. Dihybrid ratio will discuss in the next session. So hope you are liking the video, hope you are understanding the matter. Please do comment that why you how you are observing the video, is are you comfortable with the video or not, and of course stay home, stay safe, and keep studying. Thank you.